Imagine being bitten by a snake. Your blood pressure crashes, your muscles stop responding, your nervous system becomes overwhelmed. For most people, that's a nightmare, but for scientists, it's an opportunity. Because hidden inside some of the world's deadliest venoms are molecules so precise, so powerful, and so biologically active that they've inspired some of the most important medical breakthroughs of the last century. Today, we're exploring the fascinating story of how deadly toxins became life-saving peptides, from snakes and scorpions to coned snails and lizards. This is the incredible journey from venom to medicine. What makes venom so special? When most people think of venom, they think of poison. But venom is far more sophisticated than that. Venom is actually a highly evolved biological toolkit. Over millions of years, animals developed venoms to hunt prey, defend themselves, or compete for survival. To do that effectively, venom molecules had to become incredibly precise. They needed to target specific receptors, specific nerves, specific enzymes, specific cells, and that's exactly what many peptides do. A venomous animal can't afford to waste energy producing random molecules. Its toxins must work fast and accurately. That's why venoms became one of nature's greatest peptide libraries. Every venomous species is essentially carrying around a collection of highly specialized peptides. Scientists quickly realized that these natural molecules could teach us a lot about human biology. The first big discovery, one of the earliest breakthroughs, came from a snake. In the 1960s, researchers studying the venom of the Brazilian pit viper noticed something remarkable. The venom caused dramatic drops in blood pressure. At first glance, that sounds dangerous, and it is. But scientists wanted to understand exactly why it happened. They isolated specific peptide components from the venom. These peptides interfered with pathways involved in blood pressure regulation. This discovery transformed cardiovascular research. For the first time, researchers had powerful biological tools for studying how blood vessels function. The result was a completely new understanding of blood pressure control. And that understanding eventually led to major advances in cardiovascular medicine. What started with a deadly snake bite ended up helping millions of people worldwide. The Gila monster changes everything. One of the most famous venom-derived peptide stories involves a lizard. The Gila monster, native to the deserts of the southwestern United States and Mexico, the Gila monster is one of the few venomous lizards on Earth. Scientists studying its saliva discovered a peptide called exendin-4. At first, nobody expected it to become famous, but researchers noticed something unusual. Exendin-4 interacted with the same receptor system as GLP-1. GLP-1 is a naturally occurring peptide involved in blood sugar regulation and appetite control. The challenge with natural GLP-1 was that it disappeared quickly in the body. Exendin-4 lasted much longer. That discovery sparked years of research. Suddenly, scientists had a template for designing long-lasting GLP-1 peptides. Today, the GLP-1 family represents one of the most influential peptide categories ever developed. And it all started with a venomous desert lizard. The coned snail masterpiece. If there were an award for the most impressive venom delivery system on Earth, coned snails would be serious contenders. These beautiful marine snails look harmless, but they hunt fish using a harpoon-like tooth loaded with venom. Their venom contains hundreds of peptide toxins. Each toxin has a specific purpose. Some block pain signals, some paralyze muscles, others interfere with nerve communication. Researchers studying coned snail venom discovered peptides capable of targeting pain pathways with incredible precision. Unlike many traditional approaches to pain management, these peptides worked through entirely different mechanisms. This opened new possibilities for treating severe chronic pain. The coned snail became one of the greatest examples of how nature can solve problems that scientists struggle to solve in the laboratory. Scorpions and their biological toolkit. Scorpions have existed for over 400 million years. That's older than the dinosaurs. Over that immense span of time, their venoms evolved into highly specialized collections of peptides. Many scorpion peptides target ion channels. These tiny molecular gates control electrical activity in cells. They influence muscle contractions, nerve communication, heart rhythms, brain signaling. Scientists realized these peptides could become valuable research tools. By studying how venom peptides interact with ion channels, researchers gained deeper insight into neurological disorders, autoimmune conditions, and cardiac function. Scorpion venom became more than a defense mechanism. It became a biological instruction manual. Why venom peptides are so valuable, you might wonder. Why not just create peptides from scratch? Why spend time studying venom? The answer is evolution. Nature has been conducting experiments for hundreds of millions of years. Every successful venom peptide survived an incredibly demanding selection process. Only the most effective molecules remained. That means venom peptides often possess qualities scientists want. High specificity, strong receptor binding, unique mechanisms, exceptional biological activity. Instead of starting from zero, researchers can use nature's designs as blueprints. Sometimes they use the peptide directly. Other times they modify it. Either way, evolution provides a massive head start. The future of toxin-derived peptides. We've only explored a tiny fraction of Earth's venomous species. Scientists estimate there may be millions of undiscovered bioactive compounds still waiting to be studied. Every spider, snake, scorpion, jellyfish, centipede could potentially contain peptides unlike anything we've seen before. Modern technologies are accelerating this search. Artificial intelligence, genomic sequencing, advanced peptide synthesis, machine learning. These tools allow researchers to identify promising venom peptides faster than ever. The next major breakthrough may already exist inside a creature most people have never heard of. The story of venom-derived peptides is one of the most fascinating examples of turning danger into opportunity. What began as nature's weapons became tools for discovery. What evolved to immobilize prey helped scientists unlock entirely new areas of biology. From deadly snakes to venomous lizards to coned snails hidden beneath ocean waves. These remarkable creatures have reshaped modern peptide science. And perhaps the most exciting part, we've probably only scratched the surface. The next revolutionary peptide may already be waiting somewhere in nature. We just haven't found it yet. If you enjoyed this deep dive into peptide science, subscribe and turn on notifications. Because next time, we're exploring another incredible peptide story. The peptide that came from a snake and changed blood pressure research forever. See you in the next video.